Welcome back. This is Lecture 28, Total Plate Count and Aerobic Colony Count. Back in Lecture 24, we split every microbiology method into two questions: how many, and which ones. Today's lecture is the how many method itself. It is the single most run test in microbiology QC, and honestly, it is the method that most of the other quantitative tests in this level are built on top of in one way or another. By the end of this lecture, you will be able to run the entire process from raw sample to final reported number. Here is what we will cover today. Number 1, you will understand exactly what total plate count and aerobic colony count measure, and what they deliberately do not tell you. Number 2, you will be able to explain why serial dilution is necessary, and calculate a dilution factor correctly. Number 3, you will know the difference between the pour plate and spread plate methods, and when each one gets used. Number 4. You will understand the 25 to 250 countable range rule, and how to handle plates that fall outside of it. Number 5. You will be able to perform the CFU per gram or CFU per milliliter calculation correctly, from a raw colony count all the way through to a final reported result. Let's start with what TPC and ACC actually measure, because the names themselves are a little bit interchangeable in practice. Total plate count and aerobic colony count are really two names for essentially the same test: a count of every viable, aerobic organism in a sample, regardless of species. It is the general bioburden test that most other quantitative methods in this level are variations of. Here is what it tells you. Overall microbial load, expressed as CFU per gram or per milliliter. Whether a product is trending toward a specification limit over time. And whether your sanitation and process control are working, in general terms. And here is what it does not tell you. Which species are present? That is the which ones question we set up back in Lecture 24. Whether a specific pathogen, like Salmonella or Listeria, is actually present. And anything at all about organisms that cannot grow aerobically on this particular medium. I want to be very direct about this. A passing TPC is not a safety clearance. Recall Lecture 27. Every single organism in the Big Six can be present on a product that has a perfectly acceptable TPC result. Keep that in mind for the rest of this lecture. Let's get into the actual method, starting with why you have to dilute the sample first. A real sample often contains far too many organisms to count directly. If you tried to plate it undiluted, you would just get a solid sheet of overlapping growth with no way to count individual colonies. Serial dilution solves this by spreading that population out across a series of tubes, each one 10 times more dilute than the last, until at least one of those dilutions lands in a range you can actually count. Look at the diagram on this slide. Each step transfers 1 milliliter of the previous tube into 9 milliliters of sterile diluent, a 1 to 10 transfer, every single time. After 5 of these transfers, the original population has been diluted 100,000-fold. And here is a practical point: you plate several of these dilutions in parallel, not just one, because you rarely know the population in advance. Plating a spread of dilutions is what guarantees at least one of them lands where you need it. Now let's talk about plating itself. There are two standard methods, pour plate and spread plate, and they answer the same question in slightly different ways. Pour plate. You pipette 1 milliliter of the diluted sample directly into an empty sterile petri dish. Then molten agar, cooled to about 45 degrees Celsius, is poured on top and mixed in by gentle swirling. Colonies end up growing both on the surface and embedded within the agar itself. This is the traditional method behind most FDA BAM procedures. Spread plate. Here, the agar is already poured and solidified in the dish before you even begin. A small volume, usually just 0.1 milliliter, is pipetted onto the surface and spread evenly using a sterile spreader. Colonies grow only on the surface, which actually makes them easier to isolate for any follow-up testing you might need to do. This method is preferred whenever colonies may need to be picked later for further identification. Both methods are answering the exact same underlying question. The choice really comes down to whether you need colonies embedded and protected within the agar, or exposed on the surface and easy to pick off individually. Now, incubation. And I want you to recall the growth curve from Lecture 25 here, because it directly explains why these conditions are fixed the way they are. Medium, plate count Agar, a general-purpose, non-selective medium, chosen specifically because it supports growth of the widest possible range of aerobic organisms. Temperature, 35 degrees Celsius, plus or minus 1 degree, under the FDA BAM method, though some methods use 30 degrees Celsius instead. This temperature is where mesophilic organisms, the ones most relevant to food and pharma QC, grow reliably. Duration, 48 hours, plus or minus 2 hours. Long enough to let slow-growing organisms show up, but short enough to avoid overgrowth or die off, distorting your count. Atmosphere, aerobic, meaning ambient air. This is true by definition. This method simply does not capture anaerobic organisms at all, which is a limitation worth remembering. And orientation, plates are incubated inverted, lid facing down. This prevents condensation from dripping onto the agar surface and smearing your colonies together. Change any one of these variables, temperature or time especially, and you change which organisms grow and how large their colonies get, which changes your final count. That is exactly why these conditions are validated and fixed rather than left to judgment. Now let's talk about counting, and this is where the 25 to 250 rule comes in. Only plates with 25 to 250 colonies are considered statistically reliable. Look at the 3 plates on this slide. Fewer than 25 colonies. This gets reported as an estimated count, because a single stray colony has an outsized effect on the result when the total number is that low. The ideal range, 25 to 250 colonies. This is statistically reliable, and it is the plate you actually count and report from. And too many, more than 250 colonies, what we call TNDC, too numerous to count. At this density, colonies overlap and compete with each other for nutrients, which actually undercounts the true population rather than overcounting it. This is exactly why you plate multiple dilutions in parallel, as we discussed a couple of slides ago. You are fishing for the one dilution that lands in that reliable middle range. Now let's actually do the math, from a raw colony count to a final reported result. The formula is CFU per gram equals the number of colonies counted, divided by the dilution factor multiplied by the volume plated. Let's work through a real example. The 10 to the negative 4 dilution plate shows 137 colonies, which falls within our 25 to 250 countable range, so this is the plate we use. Step 1. The dilution factor at 10 to the negative 4 is 1 divided by 10,000. The volume plated was 1 milliliter. Step 2. CFU per gram equals 137 divided by 0.0001 times 1, which equals 137 divided by 0.0001, which equals 1,370,000. Step 3. The reported result is 1.4 times 10 to the 6th CFU per gram. Rounded to two significant figures, following standard reporting convention. Why round to two significant figures? The plate count method carries real statistical uncertainty. Reporting 1,370,000 implies a level of precision that this method simply cannot deliver. Rounding to 1.4 times 10 to the 6th reports what you actually know, honestly, without overstating your confidence in the exact figure. Now let's look at what this actually looks like in practice. Take a look at the 3 photos on this slide. First, a countable plate, a poor plate showing a well-spaced, countable colony range, roughly 25 to 250 CFU. This is your target result. Second, a TNDC plate, confluent, overlapping growth where individual colonies can no longer be distinguished from each other. This is what happens when you plate an insufficiently dilute sample. And third, spreader contamination, a chain of colonies caused by a dragged inoculating loop, rather than genuinely separate organisms. This is important to recognize because a spreader colony gets excluded from your count entirely. It is not a real independent colony forming unit. Once you have seen all 3 of these side by side, reading a real plate on the bench becomes much more intuitive than working from a written description alone. Before we close, let's walk through where TPC results commonly go wrong. The first mistake is counting a plate outside the 25 to 250 range as if it were reliable. reported as an estimated count instead. Treating an out-of-range plate as precise data misrepresents the method's actual accuracy. The second mistake is choosing only one dilution to plate. You rarely know the population in advance, so plate several dilutions in parallel to make sure at least one lands in range. The third mistake is counting a spreader colony as a single CFU. A dragged inoculating loop creates a chain of merged colonies from what is really just one organism, and it needs to be excluded from your count entirely. The fourth mistake is reporting excessive significant figures. Writing 1,370,000 CFU per gram overstates the method's precision. Round to two significant figures, matching what the method can actually support. And the fifth mistake is treating a passing TPC as a safety clearance. TPC measures general bioburden only. It says absolutely nothing about the presence of any specific pathogen. Let's bring this lecture to a close with the key takeaways. Number 1. TPC and ACC count every viable aerobic organism, regardless of species. It is the general bioburden test that the rest of this level builds from. Number 2. Serial dilution spreads a dense population out until at least one dilution lands in a countable range. Always plate several dilutions in parallel. Number 3. Pour plate embeds colonies in agar. Spread plate keeps them on the surface for easier picking. Both methods answer the same underlying question. Number 4. Incubation is fixed at 35 degrees Celsius, plus or minus 1 degree, for 48 hours, plus or minus 2. Change either variable, and you change the result. Number 5. Only plates with 25 to 250 colonies are statistically reliable. Outside that range, report an estimated count instead. Number 6. A passing TPC is not a safety clearance. It says nothing about whether a specific pathogen, like one from the Big Six, is actually present. That is Lecture 28 complete. You can now run and read a total plate count. You know how to dilute, plate, incubate, count, and calculate a TPC result from start to finish, and just as importantly, you know exactly what that result can and cannot tell you about product safety. Up next is Lecture 29, Yeast and Mold Testing. That lecture applies almost this exact same quantitative framework, dilution, plating, counting, calculation, to fungi instead of bacteria, with a few important differences we will walk through. Take a look at the roadmap on this slide. Lectures 24 through 27 are done. We are wrapping up Lecture 28 right now, and 7 more lectures remain in this level. If this lecture helped you, please give it a like. Subscribe for new lectures every week. And drop your questions in the comments, they get read and answered. See you in Lecture 29.